All right. Hello, everyone. I'm Rob Quinn. I'm here to talk to you today about primarily a story about cystic fibrosis. And I actually don't have a single equation in this talk, which will please some of you and perhaps displease others. But primarily, what I've been doing with Dr. Forrest Rower, who has visited the Newton Institute for actually not sure quite how long, but he was here previously, is applying methods for microbial ecology to disease context and a chronic polymicrobial infection of cystic fibrosis. So what is cystic fibrosis? Basically, it's a genetic disease. The inherent cause is genetic because there's a gene called CFTR that encodes a protein, a pretty large protein, that sits in the epithelium of a number of tissues, especially the airway. Or it's very important in the airway, in the lungs. It also is present in the gut, as well as the male reproductive tract and the pancreas. So CF presents as a number of different complications, but principally, one of the major complications is problems with lung function. So the responsibility of this gene, this protein CFTR, is to transport negatively charged ions. But in the case of CF, if you have mutations in that gene, it doesn't function properly. And there's a wide number of different mutations that can result in various degrees of cystic fibrosis. But ultimately, it's the inability to transport chloride and bicarbonate across the epithelium. So what happens is you get a disrupted, osmotic balance across the surface of the cell, leading to a dehydrated mucosal surface, which causes the buildup of thick, sticky mucus. So this is a genetic disease, and upon birth, this mucus starts to build and is subsequently infected by opportunistic pathogens. So it becomes a chronic infection, and this disease really is a chronic polymicrobial infection because as the patient ages and goes through time, they develop this very complex community of microbes that exists in the lungs, in the thin bronchioles, deep in the lungs. Some of the bacteria that infect CF patients are some of the best known pathogens that we studied, including Pseudomonas aeruginosa, Staphylococcus aureus, Genotrophomonas, Maltophilia, and others. But another important characteristic of the disease is the need for antibiotics. So these patients are basically taking antibiotics almost constantly from birth. And they're said to take kilograms upon kilograms of antibiotics through their lifetime. And some of the most antibiotic-resistant bacteria on the planet are known to exist in CF lungs. It creates a pretty challenging for treatment. So for my postdoc work, primarily has focused around a significant characteristic of cystic fibrosis called pulmonary exacerbations. Basically just that, where the disease all of a sudden gets much worse. Particularly, their symptoms increase. They can present to doctors with a number of increased symptoms that is somewhat arbitrarily defined as an exacerbation, where the patient all of a sudden gets very sick. And you can kind of see that in this plot here if you follow what is called the FEV1, which is a measure of lung function in a person through time, where this is the percent of a normal person. So normally this CF patient is around 60, maybe 62%, but all of a sudden you get this drop in their lung function periodically that happens. That's an exacerbation, and they're diagnosed based upon that. The causes of these are mostly unknown. So we don't really know what are the drivers of these periodic decreases in lung function, and they certainly are associated with poor patient outcome. The more frequently they have them, the more likely they are to die at a younger age. Even the treatments of these events are often unsuccessful, where the doctors are kind of not know quite what to do, or they'll just know that they need to provide antibiotics. We're not sure which ones. Oh, well, this antibiotic worked last time. Let's try it again. Okay, it didn't work. Now we have to change. So they're clearly a poorly understood drivers of these disease events that ultimately lead to poor outcomes of patients. So another thing I'll touch on is referred to as the clinical culture bias. So most of the microbial community experts in the audience will appreciate that there is a large diversity of microbes out there that we cannot culture. But this concept hasn't really translated well to the clinical environment. So we actually know from a number of well-done 16S ribosomal RNA studies that the CF lung is much more diverse than first appreciated. And just to give you an example of that, this is a typical microbiome of a patient in San Diego. Describing a typical microbiome is somewhat ridiculous because they often change a lot between people. But this is just kind of a theoretical example. So the bacteria that are grown on these clinical culture plates, they look for particular pathogens that are known of this disease: Pseudomonas, Staphylococcus, and Genotrophomonas. What I'm showing here in this microbiome, there's a lot of bacteria that they don't even detect. So Pseudomonas in many patients constitutes a large proportion of the bacteria in their lungs. But we also have other commonly found bacteria such as Streptococcus and Granula catella, which are very closely related. They grow well on these agar plates in these clinical media, but they're ignored by the clinic. They're considered oral contamination and basically discarded. Another example is Rothia, a bacterium emerging pathogen, they say, in CF that contributes sometimes upwards of 80% of the microbiome reads in our patients are Rothia. This bacterium is easily outcompeted on agar plates and doesn't grow very well. Finally, anaerobes. Pretty much all patients have some degree of the presence of anaerobes in their lungs, which is somewhat counterintuitive, but I'll talk about that later. These bacteria do not even grow at all on these clinical media. So really, you're only getting a small proportion of true infection of the patients, likely explaining some of the difficulties with the ineffectiveness of the treatments. So take a step back for a second and kind of give an analogy of maybe the history of this disease, and you can determine whether it's somewhat of a ridiculous analogy, but looking at microbial ecology in CF. So imagine that we were going to go and study the African savanna. As a biologist or an ecologist, we want to study the communities of organisms and plants and things that exist on the African savanna. Going there, you might, of course, want to study the lions. So in the case of CF, we know that it's a polymicrobial community in the lung. So you go to the African savanna and you may, sure enough, study the lions. And that certainly has been done for cystic fibrosis. So Pseudomonas aeruginosa, which you actually heard about in the previous talk, is most certainly an important pathogen in this disease and has been studied extensively in the context of CF and a number of other contexts. But in reality, what this is kind of like, kind of compared to CF, is going to the African savanna and not just studying the lions, but only studying the lions, and even going as far as studying the lion's teeth and learning everything you possibly could about how this lion's teeth are used to tear apart a zebra or something. In some ways, this is kind of what's happened in the history of CF microbiology. Pseudomonas aeruginosa, because of available genetic techniques and its easy growth, we know an extensive amount about how Pseudomonas aeruginosa can cause disease. But really, what is that telling us about this environment, the African savanna, or in this case, the CF lung? Little. There's good reasons for this, because basically the tools weren't available to study complex microbial communities at once, which is what we're all kind of here to try to understand. But now we have these tools, metagenomics, high-throughput sequencing, metabolomics, things like that. We can approach these complex systems and really start to get at this ecosystem that we knew existed but couldn't study very well. So kind of our working hypothesis about primarily revolving around CF microbial ecology and exacerbations was a paper published before I arrived in the Rower lab called the Climax and Attack Model, taking the word directly from macroorganism ecology. Basically, it describes the existence or the potential existence of two different microbial communities in the CF lung. A climax community, which represents bacteria that are probably always there and present and stable at the times of disease, and the attack community, which represents these exacerbations. The belief was that these organisms are transient, they colonize or get in, sometimes they even represent viruses, cause exacerbations, but ultimately you get back to this climax community. So I'll touch back to that after kind of going through some of our results. So one of the first things that I wanted to look at was metagenomics. So there's been a very good description now of what bacteria exist in CF lungs based on 16S. Really what we want to know is what genes are they bringing with them, how are they surviving, and how are they persisting. So what we can do then is sequence the metagenomes and look for particular pathways belonging to core microbial physiology. And what I basically found, kind of shown in this plot, which is the abundance of certain KEGG pathways in a CF metagenome compared to a healthy saliva. And then the size of the bubble is the abundance of that pathway in the transcriptome. Essentially, the CF microbial community is enriched in amino acid metabolism, folate biosynthesis, lipoic acid metabolism, oxidative phosphorylation, and fermentation. So a number of things you'd expect to find, but also we found some things that we didn't expect to. So really trying to just understand what is the core physiology of this microbial community and how do they get their energy and food, basically. Another thing that we can do is look a little further into these databases, not just looking at pathways, but particular enzymes that we know are important for microbial physiology. We just saw a redox tower that you'd set up. This is very similar to what I'm about to show, where we look for expression and the presence of genes in the sequence data to terminal electron receptors that we know are important for microbial respiration. Cytochrome C oxidase, cytochrome C oxidase is CBB type, which is a microaerophilic form, nitrate reductase, iron, and a number of other commonly used heterotrophic respiratory mechanisms. So what we can do then is looking at the abundances of these genes in the context of the electrical potential of the reductant is to start to build a kind of electron tower and start to understand how these microbes might set up in the lung. So we see a lot of aerobic respiration, particularly with this CBB type in the transcriptome, but then this large component of nitrate reductase presence and expression, which is primarily for denitrification and nitrate reduction, and some other inorganics as well. So what we can then do is take those sequences that hit to those particular genes and track them back to which organism they came from, and start to set up who might be doing what in this kind of theoretical electron tower. So we see here that hits for the cytochrome C oxidase mostly belong to Streptococcus, Pseudomonas, but interestingly, we see a lot of hits to this CBB-type high-affinity oxygen respiratory enzyme from Pseudomonas. And I have some evidence to support that it likely grows very well microaerophilically. And nitrate reductase contains Pseudomonas, not surprisingly, but also Raphia produced a lot of that. So ultimately, what we're doing then is starting to kind of set up niche partitioning in the CF lung using metagenomics as a hypothesis generator that we can then go test in other methods. Another thing I looked at was the CF microbial nitrogen cycle. So not unlike environmental microbes, microbes and polymicrobial infections depend on each other to recycle nutrients. Although it hasn't been applied extensively, this ecological approach to microbiology to clinical disease, really they deal with the same issues that we know very well and are very well studied in soil organisms and things like that. So this is just depicting what we know as the nitrogen cycle. And again, I can go into the metagenome data and transcriptome data and look for these particular genes or their abundances. What I see then is the, again, presence of a lot of NAP and NAR, which is nitrate reductase, lots of expression and presence of that gene, but also all of the genes of the denitrification pathway were present, and also a lot of amino acid assimilation and amino acid breakdown, indicating that we're producing ammonia from amino acids and assimilating it back into cellular material. And also DNRA by the NRFA gene with some unexpected pathway. What I didn't see, though, was the cycling back of this nitrogen cycle with the oxidative processes, particularly nitrification. So we postulated then that perhaps the CF microbial nitrogen cycle is incomplete. And this creates a problem where you're essentially building ammonia, and your ability to assimilate it back into cellular material probably limits your growth. So we tested that in a little culture system I'll talk about in a minute, where we measured the amount of nitrate, nitrite, and ammonia in sputum, and then the amount after growth in 48 hours of these tubes. And sure enough, we see massive amounts of ammonia built. Kind of verifies our hypotheses. So then what we can do is kind of visualize this data and what it's mean and how it's setting up what we think this ecosystem looks like in the lung. So this is a figure that basically depicts all of the information that we got from the CF metagenome data in the context of a CF bronchiole full of mucus. So likely oxygen only penetrates so far into the CF lung, and we see all these anaerobic processes going on deeper in the mucus, including sulfur reduction, fermentation, denitrification, and things that I just described. What's interesting about this is it's not all that unlike one of the first, or the first really, microbial ecology experiment ever, one of the first, the Winogradsky column. So what we think is that the CF microbial community likely sets up and stratifies very similarly that it does in soil. And this was done in about 1890, so over 100 years ago. So what we do then is set up this environment. If we want to study these microbes in the environment that we believe that they come from, let's set up more similar to the environment that we can try to reproduce. So this is what I call the WinCF model, which is the Winogradsky cystic fibrosis model. Basically, all it is are thin capillary tubes that are about 70 millimeters long and 4 millimeters in diameter, the same ones that you use to take, the doctors take blood in the doctor's office, and fill them with what's called artificial sputum media, which is basically fake sputum, our best effort to reconstitute mucus. Sputum is what's mucus coughed up from a CF patient's lung. What I do then is plug it at the bottom and leave it open to air at the top. So it kind of mimics a plugged bronchiole and sets up these stratified communities. Basically, it's meant to replicate a mucus-plugged CF bronchiole, which you can see here, which we know exists in this disease. To kind of demonstrate the importance of oxygen in stratifying these communities and demonstrate how it does in fact stratify in this system, we had a couple of studies that have looked at oxygen concentrations in CF mucus, where in the open airway you see lots of oxygen, and as soon as you penetrate into the mucus, it completely disappears. There's a lot of hot debate in the CF world about whether or not the mucus truly is anoxic, but we think it's likely is, and kind of demonstrate here, where this is the picture of a tube prior to incubation, but what's inside these tubes is called a ruthenium oxygen optode, which fluoresces different colors in the presence or absence of oxygen. Orange is when there's high oxygen, so at 0 hours, if I inoculate Pseudomonas into these tubes, we see lots of oxygen. Sure enough, after 24 hours, as you would predict, oxygen is really depleted from most of the tube and only penetrates about 3 millimeters into the environment. kind of demonstrating what we postulated would exist in this system. What we really wanted to do, though, is get information about the microbial physiology of patients quickly. What's going on in a patient's community? Can we compare samples between each other, compare samples through time? So what I did then was take numerous chemical indicators to help visualize responses of this microbial community in the tube. And these are basically everything I could grab around the lab. So a pH is a dye, it's a colorimetric indicator, just bronchosol purple and phenol red. Respiration dye, which is a tetrazolium redox dye, which is basically triggered upon respiratory processes. And then just adding Koumassy brilliant blue to help visualize cell material. And then finally, what I actually found somewhat by accident was when I came one morning to look at the tubes after incubation, they'd fallen over in the incubator and filled with gas. So if I kept them upright, the gases would escape, but if they were on their side, I'd get these gas bubbles. So now I have 4 variables that I can measure in the context of CF microbial physiology and get information from samples. What we do then is take pictures of these tubes and use the JPEG image, import it into MATLAB, and get intensities of the color of each pixel in these tubes down the length of these capillary tubes to give us quantitative information so we can start doing some measures and doing some stats. Just to give you some examples of what this WinCF system looks like, this is what happens with Pseudomonas aeruginosa pure culture. We see a large raise in the pH of its environment, where purple is a high pH, lots of respiration, and sure enough, at the microaerophilic interface, if you believe me, you can see a lot of triggering of the dye. It fills the entire tube with biofilm and does not produce gas bubbles. And what's interesting is the contrast to a sputum sample. So if we take a sample from a patient that even has Pseudomonas, we often don't get the same response. So studying these pure cultures of organisms in the context of a polymicrobial community can often give you the completely wrong answer. What we see here is a low pH in this patient's sputum sample, less respiration, less biofilm, but sometimes these gas bubbles are produced. However, this changes depending on who you are. So a number of patients have different responses. This is one individual, that's another, that's another. So we think we're seeing different microbial physiologies in different people. So it likely reflects the different microbial communities. This was in contrast to healthy person's sputum. So if I was to try to cough up sputum, inoculate into these tubes, most of the healthy people always get the same response. Low pH, virtually no respiration, no plugging, and lots of gas. So CF people were very different, but healthy people were consistently the same. Finally, I was able to get samples of an explant lung, which is basically a patient getting a lung transplant. We take small samples of the lung, homogenize it up, inoculate it into these tubes. It was not really a fun ordeal, but we get the same response as Pseudomonas aeruginosa. And that basically validates what we know, is that over time, patients basically get dominated by this pathogen, likely due to its high antibiotic resistance and ability to adapt to the lung. Oops. But what was really interesting is when I started using this WinCF model in longitudinal samples. So again, getting back to exacerbations, we want to know, we've set up this core physiology, we're starting to understand how they exist. Does this change through time? Can we find biomarkers of CF exacerbations? So this is looking at the pH and the gas production of these tubes from sputum samples taken in one patient through 2 different exacerbation events. And the arrows indicate when the antibiotic treatment was given. So the time before is basically dead red day, if you will, where the patient comes to the doctor, presents with a full exacerbation. What you see is that at that event, a drop in the pH for the microbial community and a massive increase in the amount of gas. So we see the pH goes down and gas goes up. And what was interesting was that this is completely reversed upon intravenous antibiotics, where now the pH is going up and the gas bubbles are completely removed, did not again return until the second exacerbation event. So we see the same thing twice in a single person. And if you take first-year microbiology, I teach you if you put E. coli in a phenol red broth with a little Durham tube, you get low pH and low gas bubble. So these are signatures of fermentation, classical signature of microbial fermentation. So we think that the physiology is in fact dynamic in this system. But looking a little further at this phenomenon, so I can take those same capillary tubes and sequence the 16S microbial ribosomal DNA in the tubes and look at which bacteria are present. So these are the same longitudinal samples. And I can highlight at the time of treatment, you see a lot of blue, which is Pseudomonas. Highly resistant bacterium. It likely was resistant to the treatments and then took over in this system after the antibiotics were administered. At time of stability, you see a more diverse community. A lot of Streptococcus, Pseudomonas. It seems to kind of fluctuate through time. But then at the exacerbation, you see what you might expect, fermentative anaerobes. Some of these are actually obligately fermentative. That's all they do is ferment. So you see that this system is really changing. It's building up to a fermentation and then completely wiped out by treatment and then likely sets back up again in this cycle. And that's what we think. Another question is, what's in the bubbles? We're really interested to see what's in this gas. Often the odors coming out of the gas were quite nauseous, so we were interested in what it was. Sure enough, we do GCMS, or gas chromatography mass spectrometry, with our friends at the UC Irvine. And we find that the biggest difference between a bubbling tube and a non-bubbling tube was acetaldehyde and dimethyl disulfide as well. Acetaldehyde being one of the first major breakdown products of pyruvate fermentation, basically one of the major steps in alcoholic fermentation. So if you're a microbe and you're a facultative anaerobe, for example, you get to pyruvate. If you have available electron acceptors, you will send things toward the citrate cycle to generate reducing power in ATP. But if you don't have those electron acceptors, we go to fermentation. So what we think is that this respiratory fuel that I described earlier in the talk may be somehow running out in this system, and it sends it towards fermentation, which ultimately leads to an exacerbation. Sure enough, acetaldehyde is actually one of the molecules that principally makes you feel hungover. So if the patients often describe this general feeling of malaise and they just don't feel well and they're not doing well. Perhaps it's acetaldehyde building in their lungs or in their blood even. Now, that story was only of one person. However, with help of Yanwei Lim in our lab, using my system, we're starting to see this in more people. So now we have the same event in 6 different individuals with massive gas production, but not during treatment and not during stability. So we're starting to think that it isn't just characteristic of one person, but it is, in fact, possibly a ubiquitous characteristic of CF disease. So what we think this can do then is kind of revise the climax and attack model. It was originally thought of as a colonization event. It may, in fact, be. We think that really what we're talking about is the core physiology of these two communities is different, where the climax community, consisting of people or bacteria like Pseudomonas aeruginosa, raise the pH of the environment to amino acid metabolism, long evolved microbes, highly antibiotic resistant. But the attack community is fermentative, and these fermentative oral anaerobes, such as Prevotella and Vianella that we saw in my tubes. Finally, if you ferment, you lower the pH of the system, leading to a large inflammatory response. It also lowers the pH of the environment, perhaps sets up a kind of a unstable state that they get stuck in, and subsequently antibiotics are required. So kind of shifting attention here, when we were thinking about this system and what we've figured out or what we believe is going on in this, it's really led us to think about chemistry. That fundamentally microbes are driven by and directly influence their environmental chemistry. So in San Diego, there's a lot of opportunity for a collaboration. So I was basically sent to Peter Dorstein's lab at UCSD to work on metabolomics of the CF lung. So using the same samples, the same approach, the same questions, but with metabolomics data. The tool that I've been using is LC-MS-MS, liquid chromatography tandem mass spectrometry. Basically how it works is I extract different molecules from sputum samples with different solvents, and then separate those solvents based on their polarity with a HPLC. Most of you probably know what that is. And then further, as they come off the column through time, the molecules are sent into a mass spectrometer, ionized, and then basically their masses are weighed using the mass spec. What's special about this type of mass spectrometry is it's tandem mass spectrometry, so then we can isolate these ions and break them apart. So we separate them by their polarity, measure their mass, and then break them apart and measure the mass of their pieces, giving us this MS2, basically the size of the pieces of the molecule. So metabolomics, unfortunately, has really lagged behind the progress of sequencing-based omics, probably due to the lack of available databases. So lots of people have mass spectrometers. They're highly accurate. They've been around for decades. But for some reason, no one's really been able to kind of push the field further due to the available algorithms that were so beneficial for sequencing-based omics, like BLAST. However, in the Dorstein lab, in 2012, a paper was published called Molecular Networking, which we believe will be the kind of the equivalent of BLAST in a way to help expand metabolomics to catch up. Basically how it works is as you get these multiple, and these are tens of thousands of MS2 spectra, so fragments of individual molecules, you can computationally begin to compare them pairwise against each other. So how this is done is they take one spectra and align it, much like a sequence alignment, and then compare the similarity. In this case, there are no fragments that are identical, so these molecules didn't break into similar pieces. But here, we do this alignment. In this case, we find we have matches. So it's a pretty simple concept that molecules that have similar structure break into similar pieces. Then you can use these MS2 spectra and align them and then score their similarity using something kind of arbitrarily called a cosine score. And then ultimately we get a relationship between these two spectra, or these two molecules in space. You do this for your entire data set, and you can get relationships of everything and build this molecular network where molecules that are similar cluster together. You can also highlight this network by computationally kind of asking, show me nodes or molecules that were present in CF patient A or CF patient B, or during exacerbation, during stability. So it's a new platform for visualizing metabolomics data that we hopefully will expand the field. Just kind of a plug, Peter Dorostein's database at UCSD, it's called GNPS. It's going to be publicly available very soon. Anyone does mass spectrometry. There is a video that I won't show because I know some people have to leave, so I'll just, maybe you can watch it if you want to get a link. You just go to gnps.ucsd.edu and you can find it. So back to cystic fibrosis. So one of the first things we want to do, this is really a new tool and a kind of a new frontier for us, is a simple question. What are the differences between a cystic fibrosis person and a healthy person in the case of their molecules? I took 7 CF sputum samples and 6 non-CF, and there were a number of statistical approaches in an untargeted fashion to see what molecules are different. We see about 16% of the metabolome is shared between both of them, but this large component in this Venn diagram of molecules that are exclusive to CF. These are inherently interesting to us. What was surprising, though, is kind of changed our focus, and sometimes I think you could imagine if you get into kind of a new field, it often takes you down places you wouldn't expect, is that we were really looking for microbial molecules, but they were really hard to find, very, very low abundance. So in a patient that had 80% Pseudomonas in the microbiome, known Pseudomonas molecules were only about 0.3% of the entire metabolome. Most of the molecules we could annotate, and I can show you the problem with the databases, about 80, 90% are unknown, were host and xenobiotics or antibiotics. So we're seeing things in the metabolomics data that's teaching us mostly about the host. Just to kind of reiterate that, taking a look at the microbiomes of these patients, so CF9 is here, blue is Pseudomonas. We don't see very much Pseudomonas. But then taking some ecological indices of the diversity of molecules, we see there was an inverse relationship between the diversity of the metabolome and the microbiome for CF in healthy people. So the metabolome was more diverse in CF, but the metabolome, or the microbiome, was more diverse in healthy. Likely kind of, again, verifying that there's not much of an impact in this metabolomic data from microbial molecules. Most of what sputum is made up of is from host. Oops. And I just said that. So our statistical approach to this is using random forest, which is a machine learning algorithm that's good for classification and identification of variables of importance in these data sets. Basically what we do is there's 2 approaches to this, supervised and unsupervised. Supervised being you tell the algorithm these samples are CF, these samples are healthy. Unsupervised, or you don't give any information and just compare similarity based on building these decision trees. So what you can do is a kind of example of random forest is in a supervised case, which is particularly interesting to us to compare, to determine what molecules are the biggest differences between CF and healthy people. You do what's called the supervised random forest, and it does out-of-bag error. So the algorithm will randomly take small components of your data set and leave out others, and then build these decision trees. So if you tell it that your square is healthy and circle is CF, and you take a component of your data and it runs down the decision tree but makes the wrong decision, that this data now represents a healthy, the molecules or the data that was left out of that algorithm was important for the distinction of the groups that you told it. So the more you do that 10,000 times, you get a greater likelihood of identifying molecules that are important for the distinction between your 2 groups, and builds a variable importance plot. So these molecules, if left out of the data, really contributed to making a wrong decision. Therefore, they're distinguishing the 2 groups. So looking at the metabolomic data and kind of combining annotation, what we found is that according to the random forest, CF lungs are loaded with sphingolipids. Again, this was not really what we were looking for, but you got to go with what your data tells you. Sphingolipids, such as sphingomyelin, are basically lipids that exist in our membranes, but they're specialized lipids. They're major components of lipid rafts, and some of them are components of gangliosides, or complex glycolipids on the surface of the cell. But they all contain ceramide, which is this core component of the lipid backbone containing a nitrogen. This molecule is an important cellular signal. When ceramide is produced in the cell, we get especially strong inflammatory signals. So we see that the CF lung is loaded with various types of these sphingolipids. Kind of depict this, so this is a molecular network cluster of related molecules. The squares are molecules that were identified by the random forest, this one being sphingomyelin, also another sphingomyelin by random forest. But what you see with the molecular networking is molecules that you weren't able to annotate. You can now start to get their relationships and see what they're related to and start to get their structures. Basically, this is a giant cluster of sphingolipids that we found. You look at the abundances in the CF and healthy, all of which are especially abundant in cystic fibrosis people. We can then look a little closer and kind of compare to some context. Well, sphingomyelin was extremely abundant in CF and virtually absent except for one person in non-CF people. Ceramide, again, much more abundant in CF, but a regular glycerophospholipid, so not these ceramide backbone, not bioactive lipids, were equal. So this does appear to be a CF characteristic of accumulation of these molecules. Looking in the literature, Googling sphingolipid and CF, you get a few hits. So there is some research around these molecules, particularly pertaining to CFTR, which I talked about earlier. What they believe is that CFTR may actually be directly involved in sphingolipid regulation, particularly ceramide. So if you knock CFTR out of a mouse, what they find is that the epithelium of those mice builds with sphingolipids. So we're starting to see that this actual inherent genetic defect of this disease may be resulting in what I'm seeing in the metabolomic data. So what I see, in fact, is this buildup of sphingomyelin. So perhaps what we're finding in the lab from other studies is showing that these molecules are building, and I'm finding them in the sputum en masse. Basically, the most abundant molecule in sputum was sphingomyelin. So this creates a problem because it represents a large reservoir of ceramide that probably shouldn't be there. Ceramide is an important component of our cell membranes, but too much of it can be a problem in the form of sphingomyelin. Makes us think about enzymes such as acid sphingomyelinase, which is one of the principal enzymes that convert sphingomyelin to ceramide. And ultimately, if that occurs, you get ceramide-induced hyperinflammation. So a characteristic of CF that's really kind of poorly described, but somewhat well accepted, is that the disease appears to be hyperinflamed, that the inflammatory response to infection appears to be particularly bad, more so than the infectious stimuli should maybe normally induce. Perhaps the reason for this, and there are some other people that believe this as a hypothesis, is due to the buildup of ceramides inherently in the lung, kind of acting like a loaded gun, that any inflammatory signals are especially strong because of this abundant ceramides. There's actually been phase 2 clinical trials for certain drugs against this enzyme, so this isn't really a new idea, perhaps that will help keep down the inflammation. So have some time. As I mentioned, metabolomics, we really wanted to target microbial molecules. But that was actually hard because the CF lung sputum was such a complex environment, most of which was host. But I did develop a method to look for microbial molecules that we can now apply to these samples. So what I do is take a sputum sample and then also take a pure culture of bacterial pathogens, such as Pseudomonas or Staphylococcus, extract the molecules in the same samples, and then run them under the same mass spec conditions, and then co-network the data together. So what you can do then is ask the algorithm to say, show me molecules and color them purple, ones that were found in Pseudomonas and in a CF patient. That way you're guiding your search toward the identification of microbial molecules, which is otherwise done manually and takes forever. Just to kind of depict how that works, so on the left is a molecular network. So these are all, every node represents a different molecule, and it's colored by attributes of the metadata, whether it was CF and Pseudomonas, or only healthy. So you look at these clusters, and we actually find molecules that we know are important for Pseudomonas physiology, such as phenazines. So the yellow are detected phenazines, including piocyanin, PCA, other phenazines that Pseudomonas produces. Quorum-sensing molecules we find in the mucus of the patient, such as quinolones, and also rhamnolipids, which are these bioactive lipids that can actually damage other cells and epithelial membranes. So just to kind of touch on what we found from these microbial molecules is looking specifically at the quinolones, we see somewhat not what we would expect. So in the red are shared mass between Pseudomonas and a CF patient. And we can see the different quinolones are present, but the PQS quinolone is not red. So it was not found in the patient. It was only found in Pseudomonas strains. So you grow Pseudomonas on an agar plate, and PQS actually stands for the Pseudomonas quinolone signal. It's the best studied of all these quinolones, but we didn't see it in the patient. But you can see all these other different chemical structures with this networking, which is pretty cool. What was the most abundant was HHQ, a different quinolone, which is actually the precursor of PQS. So the most abundant molecule is the precursor of the most notorious one. Why is PQS not there? What we think we might be seeing is that it's likely indicating anoxic growth of Pseudomonas. The final step in quinolone synthesis is the production of PQS from NADH and oxygen. If you're growing anaerobically, oxygen is not readily available. Likely we see the buildup of HHQ, kind of validating what we think we see in this environment. So finally, the question with this metabolomics data, do these molecules change during exacerbation? And the answer is yes, they do. So somewhat surprisingly, it's actually been really difficult to see changes in community profiles in CF lungs. My capillary tube system demonstrated it, but most directly 16S profiles don't often see much change as a patient goes through an exacerbation. But if we look at the chemistry, we are seeing that there are major differences. So a green sample, this is a random forest, an unsupervised one. This basically just measures the similarity of the samples, projects them with multidimensional scaling. And you can see that a stable sample, 89 days prior to the exacerbation, is here in the plot. 43 days we move. Then the exacerbation, the vector gets sent completely different direction, indicating a major change in the metabolome and the chemistry of the lung. Treatment in the purple sends things back the other way. First couple days are relatively similar. Then we get a very big change after 3 days in this plot. What we saw is following these metabolomes through time is they actually fluctuate back and forth. Whoops. They actually fluctuate back and forth in this plot, about 3-day intervals. And finally, at the stable state, 45 days later, you're more back to where you were to begin with. What we find is looking at the molecules we know we can look for is we see major spikes in these molecules, likely explaining the fluctuations in this metabolic data. So what we think we're seeing is that at the exacerbation, the sphingolipids, particularly sphingomyelin, is increased, and we get spiking of these inflammatory signals through time, perhaps kind of validating what we think might be going on with this hyperinflammatory response. But what's interesting is that this only really appears to be occurring in a particular component of sphingolipid metabolism. So ceramide, kind of the hub of this metabolic pathway, gangliosides, which are non-bioactive or more sugar-containing lipids for cell recognition, do not really fluctuate, which is in the purple line. But ceramide and sphingomyelin appear to be going up and down. You can see that in the green here, likely indicating that it really is going on at the level of the conversion between these two molecules. So ultimately, what does the metabolomic data taught us about cystic fibrosis? Well, we kind of have this picture now of Pseudomonas microcolonies growing in the lung, producing these notorious compounds that we can find, including phenazines, which induce reactive oxygen species and damage the tissue, rhamnolipids directly damage tissue, and all the while, sphingolipids accumulating due to the inherent mutations of CFTR, finding them in the sputum, the potential for conversion to ceramide inducing hyperinflammation. So to link it all together, we think now about our model about fermentation driving exacerbations and the inherent problem of this buildup of sphingolipids. So we have CFTR mutations. It leads to mucus-plugged airways, characteristic of CF, induces microbial colonization. Normally, and principally, most of the bugs are exceptional respirers, including Pseudomonas. But if they run out of electron acceptors, we're not sure why. We're trying to figure out what might be triggers of this happening. Perhaps a part of the lung gets especially plugged off and becomes very, very anaerobic, not sure, or nitrate perhaps gets depleted, and it leads to microbial fermentation, gas production, and ultimately an exacerbation. But at the same time, this fermentative response lowers the pH of the lung, and leading back to this enzyme that we know is one of the principal enzymes for the conversion of sphingolipids, which are accumulating due to CFTR mutations, to ceramide. So the exacerbations are essentially driven by microbes, but the genetic defect in the disease makes them especially harmful. That is our new kind of model system about what we think is going on longitudinally in these patients. And part of the reason I'm here, hopefully, is to work with people in the audience or anyone else that can help us think about how to model these systems and what we might need for quantitative input to try to perhaps predict certain threshold levels of fermentation signatures or respiratory signatures that might indicate a patient's heading to an exacerbation. With that, I'll say thank you. Everybody can still get to their Skype meetings. Professor Rower, who some of you probably met, it's a great character. Dr. Peter Dorstein, who's been really gracious, allowing me to basically come to his lab and somewhat fumble with his mass spectrometers. And Dr. Doug Conrad, who's the clinician that we work with very closely at SDSU. And I'll put some palm trees in the background as to where I come from. Thanks. Questions? Yeah.
Do you think the ceramide might be an input that encourage, the human metabolites? Do you think they give preference to any type of microbiota community?
I'm not sure about that, but what we do know, just a couple slides back, is that Pseudomonas actually produces a specific enzyme called PLCH that also converts sphingomyelin to ceramide. Why it might do that, I'm not sure. But we think that perhaps sphingomyelin maybe even be a recognized signature for Pseudomonas when it's in the lung, because it does produce this enzyme. But I guess the characteristics of these molecules is that they're very hydrophobic. So the CF sputum is in fact very hydrophobic. There's a lot of lipids in there that we see. So I guess bacteria that would survive better in that environment might be. But ceramide, we don't think it damages microbes or anything. It's a bioactive lipid that signatures inflammatory pathways in our own cells.
You said you had these rapid onsets and then offsets, I guess. The turnoff is because of treatment?
Yeah, so hopefully I didn't say they were rapid, because they're often not. They seem to be kind of progressive, where the patient starts to feel like crap, and then they go to the doctor, and he says, okay, you're try this antibiotic, and didn't get any better, and then the lung function goes down, and then, okay, you're on an exacerbation, hospitalized and treated. But yeah, certainly, again, so we think that the treatment is kind of hit or miss, right? And to really extrapolate the hypothesis, that the reason that the antibiotics sometimes work and sometimes don't is whether or not they kill the anaerobes. Some of the antibiotics they treat, including tobramycin, don't really have any coverage of anaerobes. The clinic doesn't report anaerobes. So the doctors don't have that information to subsequently treat. But in this case, what I talked about, we did see a direct response where within days, the community was knocked down, but the patient still feels like crap through those 14 days of treatment, probably due to these massive inflammatory responses that are induced and probably going on in the lung as it builds. So it isn't, I didn't mean to say that it was a sudden event, because it builds somewhat slowly, gets really bad, and they feel like crap for even a month at a time. But they periodically occur depending on how old the patient is, 1 to 2 to maybe even 4 times a year.
Some of your data looked like it was fairly sudden.
Yeah, so that's in the microbial physiology, which I think is an important finding in this study, is that things are changing by the bacteria really quickly. But likely that's the effect on the patient is probably more, is a lot more going on. That makes sense.
So the feeling crap is due to the hyperinflammation?
Whoops, there it goes. I think it's due to a lot of things. I mean, it could even be due to fermentation itself, where the production of things like acetaldehyde and another molecule in a paper that we published, the first author is Katrine Weitzen, 2,3-butane-dione was another molecule commonly found, a fermentation product that is the, I don't know how to do this.
I mean, you could test this, for instance, and if it would be due to this, you could, like treatment with steroids should help.
Yeah, so CF patients are treated with steroids, but the availability of steroid treatments is pretty limited. A lot of the clinicians kind of complain about that. They wish they could use more steroids. But this disease is relatively specific, so everything has to go through a clinical trial just for CF. But yeah, I think it probably is true that the inflammation is majorly damaging the tissue and leading to progressive lung decline because of its reactive oxygen species and things that inflammatory processes produce. But perhaps the microbes themselves are also directly contributing to the damage in the lung.
And the step before you say it's acid dependent, so the conversion to the ceramide?
Yeah, so that acid sphingomyelinase, its optimal activity, hopefully I don't misquote this, is about 5.4. So it primarily exists in lysosomes. It's found other places. So we think that and the pH in those tubes was below 5. So maybe even in microenvironments, the pH can get that low and favor the conversion of this molecule that's normally regulated by pH.
So could you make the lung environment more basic or something?
That's exactly the idea. So, the major problem that we all face in tackling microbial infections is antibiotic resistance. We have to figure out better ways to do this. So at least in the context of polymicrobial infections, I think we can cut things off in the core physiology. So if I showed you that electron tower, let's figure out where nitrate's coming from and chelate nitrate. Some of those ideas are gone on. A lot of work's been done around iron in CF. The CF lung's loaded with iron. And they actually use chelators to help bind up the iron and keep the microbes from using it. So I think those kind of approaches we might start to see will be more commonly used in treatment of infectious disease because they're just so good at getting genes, but their core physiology matters and they can't transport that as easily. Mm-hmm.
This isn't really a microbiology question, but is there some kind of evolutionary story or adaptive story for why these mutations that are so harmful in CF patients that cause cystic fibrosis, why they exist?
That's a good question. So there's some theories that are obviously tough to test because it's a historical story, but the principal kind of hot topic now is cholera. So if you're homozygous, it's a classic Mendelian recessive, autosomal recessive. So if you're a heterozygous, you have somewhat lower activity of this. So if you get cholera in your gut, which produces massive influx of water into your gut, then you have terrible diarrhea and you die. But if you have somewhat lowered function of CFTR, you likely survive that better. You have less massive outflux of water, makes sense. The other idea is a salmonella infection. So salmonella typhimurium or typhus, and this is all believed to be around the Middle Ages where there's like a bottleneck die-off of people and it's selected for heterozygotes. Whereas salmonella typhimurium, its receptor for cells is CFTR. So if you had mutations in CFTR, then salmonella wouldn't be able to infect you. You would survive that. But it is a lethal mutation. The median age is about 40 now. 20 years ago, it was like 18. So Y has such a lethal mutation. It's probably really strong selective pressures, they believe, around the Middle Ages.
You said that there's some membrane transport or something?
Yeah.
So that's expressed in the male reproductive tract? So
Yeah,
do that?
but not the female. So the males are sterile, I believe.
Oh, so male homozygous is sterile.
Right, so that has a huge effect. Yeah, I talked about that recently with another guy. So yeah, the males are sterile, my understanding, but females are not. They can have children. Okay, thank you.